Breast cancer is a type of cancer that develops in the breast tissue. It is one of the most common types of cancer among women worldwide, and it can also occur in men, although this is much less common. Breast cancer can develop in different parts of the breast, including the ducts that carry milk to the nipple or the lobules that produce milk. Breast cancer is a major public health concern, affecting millions of people around the world. According to the World Health Organization, breast cancer is the most common cancer in women worldwide, with an estimated 2.3 million new cases diagnosed in 2020 alone. This represents about 11.7% of all new cancer cases diagnosed in women globally. In the United States, breast cancer is the second most common cancer among women, after skin cancer. It is estimated that about 1 in 8 women in the U.S. will develop breast cancer at some point in their lifetime. According to the American Cancer Society, it is estimated that in 2023, there will be about 297,790 new cases of invasive breast cancer diagnosed in women in the U.S., as well as about 55,270 new cases of non-invasive breast cancer. Although breast cancer is more common in women, men can also develop the disease. According to the American Cancer Society, it is estimated that about 2,800 new cases of invasive breast cancer will be diagnosed in men in the U.S. in 2023.
Breasts are made up of fatty tissue. They contain small chambers called lobules where breast milk is made. The milk travels through tiny channels called ducts to reach the nipples. Lymph nodes are located inside your breasts and under your arms. They are connected by lymph vessels, and they help your body fight disease. Breast cancer occurs when healthy breast cells become abnormal, grow out of control, and form tumors. Breast cancer can sometimes spread to other parts of the body. About 1 in 8 women have a lifetime risk of getting breast cancer. For men, the risk is 1 in 1,000. Breast cancer that develops inside the milk ducts is called ductal carcinoma in situ, or DCIS. When DCIS spreads into surrounding tissue, it is called invasive ductal carcinoma, or IDC. Abnormal cells can develop in the lobules of the breast. They are called lobular carcinoma in situ, or LCIS, and are not cancer. When these cells spread into surrounding tissue, they become a cancer called invasive lobular carcinoma. Doctors classify breast cancers based on how they behave. This includes non-invasive, cancer that has not spread outside the tissue where it began; invasive, cancer that has spread and invaded healthy tissue; recurrent, cancer that returns after it was treated; and metastatic, cancer that has spread from the area where it started to other areas of the body. Common types of breast cancer are invasive ductal carcinoma and invasive lobular cancer. Less common types of breast cancer include inflammatory breast cancer, tubular breast cancer, colloid breast cancer, and metaplastic breast cancer. Triple-negative breast cancer means the tumor has no estrogen, progesterone, or HER2/neu protein receptors on its surface. Triple-negative disease may have a worse outcome and often requires more intensive treatment. Risk factors for breast cancer include older age, dense breasts, having breast cancer before or having a parent or sibling who had it, having specific inherited gene mutations called BRCA1 and BRCA2, having Ashkenazi Jewish ancestry, drinking more than a small amount of alcohol, and being overweight or obese, especially after menopause.
Your risk for breast cancer increases with age. About 77% of women are over the age of 50 when they are diagnosed with breast cancer. Less than 1% are diagnosed in their 20s. If you have a close relative, a mother, sister, or daughter, who has had breast cancer, your own risk of developing the disease doubles. If 2 of these relatives have the disease, your risk increases 5 times. In many cases where breast cancer seems to run in families, a mutation in specific genes called BRCA1 and BRCA2 may contribute to the cancer. If you started menstruating before age 12, or went through menopause after age 50, your risk for breast cancer is also slightly increased. That's because your breast tissue is exposed to estrogen for longer periods of time. And finally, there is a clear link between alcohol and breast cancer. Those who have 2 to 5 alcoholic drinks daily have 1.5 times the risk for developing breast cancer as women who do not drink. Most breast changes are due to hormonal changes and are nothing to worry about. However, if you experience any of the following 5 breast cancer symptoms, see your doctor, even if they seem mild. 1. A lump in the breast or armpit. 2. Discharge from the nipple. 3. Dimpling on the breast. 4. Nipple changes, including the nipple turning inward, pulling to one side, or changing direction. Or 5. Irritation of the skin.
Screening for breast cancer can help find cancer early and save lives. Screening includes examining your own breasts, having a healthcare provider examine them, and having an X-ray of the breasts called a screening mammogram. You can check your own breasts about the same time each month. Feel each breast and armpit and look for any of the signs of breast cancer previously discussed. A diagnostic mammogram is used when a change is seen on a screening mammogram. It includes extra detailed images of the breast. A breast ultrasound is often used to determine if a suspicious lump is solid or fluid-filled. A fluid-filled lump is often benign or a cyst, while a solid lump can be cancerous or a benign fibrous growth. A breast MRI shows more detail than a mammogram and is often used for very small masses or changes in the breast that cannot be seen by mammogram. A breast biopsy is a test that removes a few cells or a small amount of tissue from the suspicious area in the breast. There are various types of biopsies. A doctor called a pathologist will analyze the breast biopsy tissue by looking at the cells under a microscope and doing specific tests. Their report will let you know if you have cancer and if it grew because of estrogen, progesterone, or HER2/neu protein on the cancer cell. The results of these tests help your doctor choose the right
Tumor
treatment.
markers are substances found in the blood, tissue, or bodily fluids that may indicate the presence of cancer. Let's explore tumor marker tests for breast cancer. 2 common tumor markers for breast cancer are CA15-3 and CA27-29. While these tests can't diagnose cancer on their own, they can help monitor cancer treatment and detect recurrence. Keep in mind that tumor marker tests can sometimes give false positive or false negative results. This is why they're used in conjunction with other diagnostic tools like mammograms, ultrasounds, and biopsies. Tumor marker tests are typically more helpful for monitoring treatment progress and detecting recurrence in patients already diagnosed with breast cancer rather than screening for cancer in healthy individuals.
Breast cancer staging is determined by the size and location of the cancer and whether it has spread to other areas in your body. Breast cancers are graded 1 through 3, with 1 being the most similar to normal, healthy cells, and 3 being the most different from normal cells and the most aggressive. Breast cancer treatment depends on the type and stage of your cancer. You will need treatment from a cancer team specializing in different areas of medicine. The main treatments include surgery, radiation, chemotherapy, hormone therapy, targeted therapy, and combination treatments. Common types of surgery for breast cancer are lumpectomy, removing the tumor and a small portion of healthy normal tissue around it; simple mastectomy, removing the entire breast; modified radical mastectomy, removing the entire breast and lymph nodes under the arm; prophylactic mastectomy, removing one or both breasts to reduce the risk of developing breast cancer; mastectomy with reconstruction at the same time as removal of the breast; sentinel lymph node biopsy, a procedure to determine if one or more lymph nodes contain cancer cells; and axillary node dissection, surgery to remove lymph nodes in the armpit region. Reconstructive surgery is a procedure done to restore the appearance of the breast after a mastectomy or large tumor removal. Radiation therapy uses radiation energy to kill cancer cells. It is mostly recommended after surgery or chemotherapy. Chemotherapy is the use of drugs to destroy cancer cells. It is given as an IV or a pill. Chemotherapy can be used to shrink a tumor before surgery, called neoadjuvant chemotherapy, kill cancer cells after surgery, called adjuvant therapy, and control metastatic breast cancer, called palliative chemotherapy. Hormone therapy is given if your tumor is positive for the hormones estrogen or progesterone. Hormone therapy is used to lower the risk of breast cancer coming back and help shrink or slow the growth of metastatic estrogen receptor-positive breast cancers. Tamoxifen and aromatase inhibitors are common hormone therapies for breast cancer. Targeted therapy uses drugs or other substances to identify and fight specific types of cancer cells with less harm to normal cells. Targeted therapies stop cancer cells from growing, identify and kill cancer cells, and increase the immune system's attack on cancer cells. HER2 is a common protein that is blocked with targeted therapy, for example, trastuzumab. Targeted therapy is often given along with chemotherapy. Metastatic breast cancer is cancer that is spread from the breast to other areas of the body. The most common places are the liver, bones, lungs, and brain. There is no cure for metastatic breast cancer, and treatment is to prolong survival. Today, people are living 5, 10, or more years with metastatic breast cancer due to advances in treatment. Metastatic breast cancer treatment may include ongoing chemotherapy, hormone therapy, targeted therapy, and new innovative medicines in clinical trials, such as immunotherapy.
Many genetic and environmental factors can cause tumors to form. For instance, UV radiation from the sun can damage DNA and other structures in melanocytes, which are the pigment-producing cells in the skin. Chronic damage to melanocytes by UV radiation leads to most cases of melanoma, which is a type of skin cancer. As melanomas grow, they can eventually spread to other sites in the body, such as the lungs and liver. The cells of the immune system are continuously monitoring our tissues. Natural killer cells, or NK cells, sense stress-associated molecules on damaged and cancerous cells. Dendritic cells, or DCs, activate cytotoxic T cells, which can then sense tumor-associated antigens using their T cell receptor and other co-receptors. Once activated, NK cells and cytotoxic T cells release perforin and granzymes. These molecules punch holes in the surface of the tumor cells, causing them to die by apoptosis. Helper T cells support these responses. They help DCs to activate cytotoxic T cells, and they produce cytokines such as interferon gamma that recruit and activate more NK cells. As the tumor evolves, genetic changes occur that can give some tumor cells a survival advantage. This means tumors are often heterogeneous. For instance, tumor cells may no longer express the molecules that are sensed by the killer immune cells. As the immune system continues to kill the tumor cells it can recognize, the cells it can't sense become more prevalent. This is immunoediting. It leads to the emergence of a tumor that can't be detected by the immune system. Some tumor cells actively suppress T cells by expressing inhibitory molecules such as PD-L1. PD-L1 binds to the PD-1 receptor on T cells and deactivates them. This is an immune checkpoint. In addition, tumor cells can attract immune cells that suppress the activity of other immune cells, thereby supporting tumor growth. These immunosuppressive cells include regulatory T cells and certain types of myeloid cells. Therefore, the tumor microenvironment is like the scene of a battle between two opposing immune responses. One side of the immune system is attacking the tumor, while the other side is helping it to grow. Scientists are developing immunotherapies to help strengthen the immune attack. One approach is adoptive T cell transfer. Cytotoxic T cells taken from patients with melanoma are selected for their ability to attack tumour cells. The best killers are infused back into the patient. Treating patients with cytokines, such as interleukin-2 and interferon alpha, can also boost the activity of anti-tumour immune cells. An alternative approach is to target the immune checkpoints. For instance, antibodies that bind to PD-1 stop this molecule from switching off cytotoxic T cells. Another immune checkpoint being targeted is CTLA-4. Blocking this molecule helps DCs to drive anti-tumour T cell responses. Not all patients will respond to these immunotherapies, and some responses will be delayed. Combining immunotherapies with chemotherapy or radiotherapy can lead to a better response in some patients. Immunotherapies can themselves be combined. For example, PD-1 and CTLA-4 blockade can improve responses when administered in combination. Activating the immune system has risks. Some patients develop harmful side effects when their immune system attacks healthy cells.
The immune system plays a vital role in detecting and fighting cancer. However, cancerous cells evade the immune system by producing proteins that cause loss of antigens and immunogenicity, or directly suppress the immune response while promoting cell division. Cancer cells express genes coding for proteins that stimulate uncontrolled cell proliferation. Protein-encoding genes are initially transcribed into mRNA in the nucleus. This mRNA moves into the cytoplasm and is translated into proteins. Gradellus has developed a novel bifunctional short hairpin RNA, bi-shRNA, able to silence the expression of any gene. Bi-shRNA induces cleavage-dependent post-transcriptional mRNA degradation, as well as cleavage-independent mRNA degradation. This mechanism enhances the blockade of protein synthesis compared with other RNAi and gene silencing mechanisms. Vigil is a novel cancer cell immunotherapy that combines bi-shRNA furin, which blocks immunosuppressive protein production, with recombinant human GM-CSF, which stimulates the immune system. First, cancer cells are harvested from the patient. The cancer cells are modified using DNA plasmids containing instructions for bi-shRNA furin and GM-CSF, and then injected back into the patient. Vigil's bi-shRNA selectively blocks furin protein production, which inhibits downstream production of the potent immunosuppressors TGF-beta1 and TGF-beta2. This increases the immune response. GM-CSF increases antigen expression on cancer cells, boosting visibility and immunogenicity. It also triggers the maturation and recruitment of antigen-presenting cells. The patient's antigen-presenting cells can recognize and process the cancer cell antigens more easily, and then migrate to lymph nodes to present these antigens to T cells, thereby activating them. These activated T cells are better equipped to orchestrate attacks on cancer cells throughout the body. They release interferon gamma, stimulating tumor apoptosis. Vigil is a personalized cancer vaccine that enables the body to heal itself. The platform can be applied to any solid tumor and has shown safety and efficacy in Ewing sarcoma and ovarian cancer.
Every day, our innate immune response protects us from cancer and infectious diseases. It's essential for our existence and is so fundamental to life that we share much of it with primitive animals such as sponges and earthworms. Adaptive immunity evolved more recently, but to be initiated, it still needs the innate immune response. Within our innate immune system, natural killer cells, or NK cells, are the major cytotoxic lymphocytes. They circulate in our blood and reside in lymph nodes and our lymphoid organs like the liver and the spleen. NK cells constantly patrol our bodies to eradicate cancer cells as they arise and provide crosstalk to the adaptive immune system. This surveillance by NK cells in healthy individuals provides a rapid response to tumour formation and is vital for the early reaction to tumorigenesis independent of T-cell immunity. So understanding tumour-specific NK cell responses is essential to maximise their therapeutic potential in cancer treatment. As resting NK cells circulate, their activation is controlled by a multi-step process requiring binding of signalling molecules on their surface by molecules expressed on tumour cells. When NK bind to cancer cells, they form a tight bond called an immune synapse, where the tumour ligands attach to their specific NK cell receptors and initiate a cascade of signals. This moves the NK cells from resting state to the point where they can kill the cancer cell. Most tumour cells provide enough of these signalling molecules to trigger NK cell killing. But tumours can evolve, and any genetic changes which lead to loss of NK activating molecules will protect them from lysis. Here you can see human NK cells interacting with and binding to ovarian cancer cells but failing to kill them. The immune synapse forms, but the absence of some of the key cancer signalling ligands prevents full activation and triggering of the NK cells. These NK-resistant cells evade NK cell killing and become more prevalent. A growing tumour ignored by these NK cells can lead to the development of cancer. Immunotherapy can overcome this resistance by providing NK cells with the additional signalling ligands which the cancer cells have lost. We've created INKmune. INKmune is a suspension of specialized inert tumour cells which are delivered intravenously. The INKmune cells circulate in the blood where they bind to resting NK cells. INKmune cells express hundreds of thousands of molecules which stimulate resting NK cells to the point where they no longer need the activating ligands which are missing from the resistant cancer cells. INKmune cells bind to NK cells via multiple different receptors, one of which is CD15. In the cell synapse, CD16 is shed from the NK cell surface, and CD3 zeta is released to bind to the intracellular domain of CD2. The CD2 receptor on the NK cell binds to CD15 on the INKmune cell, causing sequential phosphorylation of CD3 zeta, LAT, and STAT5, and leading to gamma interferon synthesis and NK activation. The primed NK cell now needs to bind fewer activating ligands on the tumour cell to be able to kill it. So cancer cells which had evaded NK cells through loss of signalling ligands are now susceptible to these primed NK cells and are killed. Here you can see INKmune primed NK cells killing the NK-resistant ovarian cancer cells which they failed to kill before. The primed NK cells form very strong bonds to the cancer cells and then induce lysis. Our research at InmuneBio focuses on the signals needed to transition NK cells from rest to lytic function, and on why some tumour cells are more resistant to NK killing. We've identified a unique signature of over 600 INKmune-induced genes in NK cells, many of which are involved in tumour cell binding and killing, as well as in NK cell proliferation and critical interactions with cells of the adaptive immune system. We expect that our increasing understanding of NK cell activation and the key molecules which play a role in tumour target recognition will lead to novel therapeutic strategies for the treatment of many different cancer types.
Our immune system is equipped with cells that form an army that help our body defend itself and fight foreign invaders. Some of these immune cells, called T cells and B cells, are programmed to get rid of foreign invaders like viruses and bacteria. T cells release toxins against the foreign invaders, and B cells make antibodies to neutralize them. We have known for some time that our immune cells can recognize cancer cells as foreign invaders and attack them by secreting special factors or weapons. How can our immune cells do this? The organs of our body are composed of billions of cells. These cells contain thousands of proteins that act like worker bees and are responsible for the function of our cells. These proteins break into small parts called peptides. Some of these travel to the surface of our cells and are presented by molecules called MHC or HLA. T cells continuously scan the surface of these cells through the T cell receptor or TCR. The TCR works as a barcode scanning machine, and the peptides presented on the cells are unique barcodes. This function of the T cells is called surveillance. If T cells recognize these peptides as normal, then the T cells move on. But if the peptides come from foreign invader proteins, then the T cells get activated to attack the invader cell with weapons to eliminate it. The T cell secretes certain proteins and chemicals called cytokines and factors. These molecules punch holes into the cells that contain the invading organisms. Proteins are your body's basic building blocks. They allow your cells to divide, rest, and function properly. But sometimes these proteins get changed or mutated. For example, smoking leads to mutations that make abnormal proteins in our lung cells. Sometimes when these proteins are changed, they stop functioning normally. This makes the cell become cancerous by making it grow and divide to become cancer. These mutated proteins are also broken down into smaller pieces and are presented as peptides on the surface of the cancer cell. These abnormal peptides are recognized by the TCR as foreign and activate the T cells to kill the cancer cells. But if the immune system can recognize and eliminate cancer, then why do people develop cancer? Sometimes the immune system fails to do its job. The immune response against cancer may not be strong enough, or cancer cells may evade the immune system. Tumors, or a group of cancer cells, can build a defense network against our immune system. These defense mechanisms can either prevent the immune army from entering the tumor or can weaken and inhibit the T cells. For example, tumors can express attack molecules on their surface that bind to the T cells and inhibit their killing activity. These molecules are called checkpoints, and examples are PD-1 and CTLA-4. The tumor can also secrete proteins, factors, and cytokines that inhibit T cells from attacking and killing cancer cells, and can also turn the T cells into friends of the tumor. These cells can then deactivate other T cells. These traitor T cells are called T regulatory cells and are the enemy of the killer T cell army. The field of immunotherapy has shown great progress toward a cure for cancer, after working at it for many years with some major recent breakthroughs. Immunotherapy is actually one of the most important revolutions in the history of medicine. What are the different types of immunotherapy? Immunotherapy functions in two primary ways: by enhancing the immune system and making them stronger, and by using drugs that help inhibit the suppressive immune environment of the tumors. One enhancement method is to create vaccines. Foreign antigens can be identified by identifying mutated proteins in the tumor. These mutated proteins, called neoantigens, can be given as a cancer vaccine. The cancer vaccine is developed to activate the patient's own T cells against the foreign antigens. Like the flu vaccine fights against the viruses, the tumor vaccine fights against the foreign tumor cells. Now that the T cells are trained by the neoantigen vaccine, they go around the body to find those specific mutated antigens and will locate them on the tumors. Another enhancement approach is adoptive T cell transfer, where T cells are taken out of the patient's body, grown in the laboratory, and educated to recognize cancer or even modified to become much stronger. These fighter cells are then transferred back into the patient. A third enhancement approach is using stimulating factors called cytokines. Cytokines are stimulating proteins like interleukin-2, 7, 12, and 15 that cause the T cells to significantly multiply and get stronger. A fourth enhancement approach uses agonist antibodies such as anti-OX40, anti-GITR, and anti-41BB, and others to cause the T cells to grow and strengthen. This allows the enhanced T cell to overwhelm the cancer cells. A second approach to immunotherapy. strategies have been developed that knock down cancer defense mechanisms. There are antibodies that neutralize the inhibitory factors and cytokines such as anti-IL-10 and anti-TGF-beta and IDO inhibitors. Some of these approaches have already been approved by the FDA, like anti-PD-1 and anti-CTLA-4. The future of immunotherapy is in combining different approaches to accomplish two goals: to create a trained army of cells to attack the cancer and to break down the defenses of these cancers by unleashing your own immune defense against cancer to improve on the currently very impressive results that immunotherapy is already providing our patients.
Every day, our body has to protect itself against pathogens in our environment, but also against cancer cells in our tissues. Immune cells play a crucial role in this protection. There are many different types of immune cells, and each cell type has its own task in protecting our body. An important cell type in this process is the T cell, also known as T lymphocyte. T cells are transported by the blood. By traveling through the walls of blood vessels, they can enter and leave tissues. When a T cell travels through the body, it will examine all the cells it encounters. To do this, the T cell uses an antenna-like structure called the T cell receptor. Using its T cell receptor, the T cell checks whether a cell is abnormal, for example, whether it has been infected by a virus, as shown here. When the T cell receptor recognizes an abnormal cell, it will attach to the cell. The receptor then gives a signal to the T cell to destroy the abnormal cell by releasing deadly molecules. T cells play a central role in cancer immunotherapy because they can recognize cancer cells as abnormal cells. The T cells can then subsequently destroy these cancer cells. In addition to the activating T cell receptors, T cells also have receptors that can restrain their function. These receptors are called checkpoints. When these checkpoints are turned on, the T cell can no longer destroy abnormal cells. Cancer cells sometimes abuse these checkpoints, thereby giving stop signals to the T cells. By doing this, the cancer cells prevent themselves from being destroyed. Immunotherapy using checkpoint inhibitors tries to prevent this manipulation by cancer cells by blocking the cancer cell's ability to trigger the stop signal in T cells. In this type of immunotherapy treatment, the patient receives an infusion with checkpoint inhibitors. The checkpoint inhibitors are then transported through the blood to the tumor site. There, the checkpoint inhibitors will prevent the cancer cells from restraining the activity of the T cells. The checkpoint inhibitor forms a barrier between the stop signal coming from the cancer cell and the T cell. As the T cell now no longer receives a stop signal, it can proceed to destroy the cancer cells. Immunotherapy using checkpoint inhibitors is a standard treatment for, amongst others, some forms of melanoma, kidney cancer, and lung cancer. Patients with other forms of cancer can in some cases receive checkpoint inhibitor immunotherapy by participating in clinical trials. The T cell can receive many different types of stop signals from cancer cells. For each of these different stop signals, different checkpoint inhibitors are being developed. Many of the immunotherapy treatments with checkpoint inhibitors are still in development. Currently, clinical trials are ongoing to test which combinations of checkpoint inhibitors are most effective against different types of cancer. Please visit our website for more information on this and other forms of immunotherapy.
Mesenchymal stem cells, originating from the bone marrow, exhibit a unique cell surface that inherently attracts them to cancer cells and sites of inflammation. NanoGhost develops an innovative therapeutic approach that harnesses the affinity of mesenchymal stem cells to cancer by preserving their membrane virtues, including their unique cell surface characteristics. By loading these nanoparticles with new therapeutic payloads, this concept pioneers a breakthrough in cancer treatment. The manufacturing process of nanoghosts begins with the proliferation of mesenchymal stem cells derived from human bone marrow. Following proliferation, MSCs are dissociated, their cytoplasm removed, and their cell membranes, with retained targeting capabilities, are repurposed to create nanoparticles known as nanoghosts. Nanoghosts can be loaded with a diverse array of payloads, ranging from small molecules to proteins and nucleic acids, including mRNA. These payloads are delivered selectively and precisely into cancer cells. Injected into the patient's bloodstream, nanoghosts travel throughout the body. With harnessed MSC characteristics and tropism to cancer, they can selectively target any tumor type, including those in the brain. Nanoghosts have a unique ability to cross the blood-brain barrier, a barrier preventing most drugs from reaching brain tumors. This capability, previously unattained, enables targeting of drugs to the brain, allowing treatment of unmet, incurable brain malignancies and metastases. Selectivity, broad oncological applicability, and penetration to the brain make nanoghost technology the next generation of anti-cancer therapy.